Hi, hello there. A very good day to you. First of all, let me introduce myself. My name is Magdline Henry Sum, and I'm an associate professor at the Institute of Health and Community Medicine, University Malaysia, Sarawak. Today, I would like to present a serological innovation that we have worked on entitled Recombinant prM and NS1 of Zika virus for serological detection. First of all, let me begin with a brief introduction to the work at hand. Firstly, a brief summary on Zika virus, and this will be subsequently followed with a brief description of the invention itself. Zika virus belongs to the genus Flavivirus of the Flaviviridae family. You may be familiar of some of the other viruses in the genus, such as dengue virus, Japanese encephalitis virus, yellow fever virus, and West Nile virus. Zika virus infection typically results in mild symptoms, including fever, rash, conjunctivitis, muscle and joint pain, malaise, and headache. However, in recent years, Zika infection has been associated with more severe symptoms, such as microcephaly in infants and also a neurological disorder, which is Guillain-Barré syndrome. The Zika virus genome, on the other hand, is a single-stranded positive sense RNA of approximately 10.8 kilobases in length. And the genome itself is organized into a single open reading frame that encodes a single polyprotein, which is subsequently cleaved into structural and non-structural proteins. The structural proteins consist of three proteins, which are the capsid protein or C protein as brief, that forms the nucleocapsid. Then the premembrane protein or the prM protein, which is cleaved into M protein during viral maturation. And the major surface protein is the envelope or E protein. The non-structural proteins, on the other hand, consist of 7 proteins, namely the NS1, the NS2A and B, NS3, NS4A and B, and NS5. These proteins are mainly involved in RNA synthesis, polyprotein processing, viral replication, assembly processes, and enzymatic activities. Why do I want to introduce the proteins to all of you? It's because the prM and the NS1 are the proteins of interest of this invention. So this brings us to the invention at hand. So what is the invention? This invention pertains to the expression of recombinant prM and NS1 proteins of the Zika virus in E. coli expression system, which is useful for detecting Zika virus antibodies for seroepidemiology study. Additionally, the invention also addresses the capability of the expressed recombinant prM and NS1 proteins to specifically identify responses to Zika virus with no cross-reactivity to other flaviviruses. Furthermore, the invention also includes specifically designed primers for the amplification of each target region and the plasmid DNA of the E. coli vector that comprises the nucleotide sequences of the target region, coding for the expression of the Zika virus prM and NS1 proteins. So, what is the necessity of this innovation? Well, serologic cross-reactivity among flaviviruses has been well documented. This is mainly due to the high degree of genetic similarities among the viruses as well as structural homology in the proteins, particularly in the major surface protein, which is the envelope or the E protein. This issue presents challenges in developing specific diagnostic tests for Zika virus infection. This issue poses an even greater challenge in determining Zika virus seroprevalence in dengue endemic areas such as Sarawak and the rest of Malaysia. Furthermore, diagnosis may often be missed due to the similar clinical presentation of other flavivirus infections such as we mentioned previously, dengue. This may be the likely reason why there are no cases of Zika virus infection have ever been reported in Sarawak. So therefore, we hope to offer a solution to this problem through this innovation. It is documented that responses to the whole viral antigen prM, which is the other structural proteins of Zika virus, do not elicit cross-reactivity with other flaviviruses. And it is also documented that detection of early infection is more sensitive when targeting NS1. Hence, this invention focuses on developing prM and NS1 antigen base for the serological detection of Zika virus infection as well as distinguishing it from other flavivirus infections. However, working with native antigens will require the use of live organism or human samples, and this will raise ethical and practical issues. Therefore, alternatively, we develop recombinant prM and NS1 proteins as substitute antigens for use in the antigen-based assay. Recombinant antigens provide a more reliable, safe, and flexible option. For this purpose, first we have to identify the target region of choice for both prM and NS1 through bioinformatic analysis, then followed by designing specific primers to amplify the target regions of both prM and NS1. Secondly, the amplified target regions were cloned into the E. coli vector using TA cloning protocol. The recombinant prM is about approximately 501 base pair, while the recombinant NS1 Zika virus is around 433 base pair. Thirdly, the recombinant plasmids were then transformed to express the recombinant proteins, and these recombinant proteins subsequently purified through column chromatography and tested in an immunoblot assay. And our result shows that both recombinant prM and recombinant NS1 successfully detect the presence of Zika virus antibody. So you may ask, how is our innovation different from what is available currently? Or you may ask, what is the novelty of our invention? Well, for a start, we have successfully engineered a recombinant Zika virus antigen that is non-infectious, that can be used as alternative to native antigen for development of diagnostic serological detection of Zika virus infection. We have also successfully shown that the antigenicity of the recombinant prM of Zika virus that does not cross-react to antibodies of other flaviviruses such as dengue. Why is this crucial? This is very important, especially in endemic areas where both viruses are co-circulating. Thirdly, the expression of the recombinant proteins in prokaryotic expression system, in our case, we were using E. coli expression system, produce stable and high yield in a short time relatively compared to eukaryotic expression system. So finally, to recap back what I've been talking about for the past few minutes, this innovation presents an overexpression of the recombinant prM and NS1 Zika virus proteins expressed in E. coli expression system, offering stability and ease of production. Additionally, the innovation presents the antigenicity of the recombinant proteins, offering an alternative to native antigens, which is an advantage because recombinant proteins are safer to handle, more stable, and simpler to produce. Furthermore, the innovation presents the ability of the recombinant prM protein to differentiate Zika virus infection from other flavivirus, especially dengue virus infection. In conclusion, this innovation presents potential antigens for development of omics technology assay, not only to detect, but also to distinguish Zika virus infection. Last but not least, I would like to thank you for your time. See you again.